All right, I'd like to start the symposium. We're going to talk, this is Dr. Howard Weiner in Boston, Brigham and Women's Hospital at the Harvard Medical School. We have an exciting symposium on innate immunity in neurologic diseases. First speaker is Francisco Quintana, who's going to talk about astrocytes. Francisco, please begin.
Thanks, Howard, and thanks to the organizers for the invitation to participate in this symposium. What I'm going to do today is actually to summarize or refer to part of our data on the role of astrocytes in CNS inflammation. And one of the common themes around this symposium is going to be a disease, multiple sclerosis, and its experimental model experimental autoimmune encephalomyelitis. As you know, as you might know, this disease is mostly driven by the distraction of this myelin here around the axons, and then that results in axon dysfunction, axonal loss, and eventually neuronal loss. So the question that many of us are trying to understand is what are the mechanisms behind the distraction of this myelin structure, with the goal of trying to understand or develop novel therapies for neurologic disorders. In the context of MS, we believe that T cells, peripheral immune cells, are actually main drivers of CNS pathology. In particular, T cells reactive with myelin epitopes are differentiated in the periphery. The mechanisms leading to that are still not clearly understood. But once those T cells get into the CNS, they are reactivated, and they can cause damage to axons and myelins. In the context of multiple sclerosis, there are multiple therapies that impact several aspects of this T-cell-driven pathology by either interfering with the activation or polarization of those T cells in the periphery, or their ability to leave the lymph nodes and make it into the CNS. This is important when we think about relapsing remitting MS, for example, because this is supposed to be one of the driving mechanisms of the disease. However, as the disease progresses, additional mechanisms take a role, become more important. And in particular, one thing that we need to keep in mind is that as these T cells make it into the CNS, they're not only going to cause damage to axons and myelin, they're also going to activate resident cells in the central nervous system, triggering mechanisms of innate immune inflammation, which are actually at the core of this symposium today, which we believe contribute to the pathology not only of the progressive stages of multiple sclerosis, but also to the pathology of other neurologic diseases. This is important because once microglia and astrocytes are activated, and this is actually a figure reproduced from a review written by Dr. Weiner. Once astrocytes and microglia get activated, then they themselves can trigger and drive additional mechanisms that result in neurodegeneration and neuroinflammation. Because of that, my lab has been interested in trying to understand the regulation of these processes with a particular focus on astrocytes. Astrocytes, as you know, are the most abundant glial cell in the CNS. They got their names because they resemble stars. And this is an important cell population that performs multiple functions in health and disease. In the context of health, in the context of homeostasis and development, astrocytes are important for the development of axons, for the function of axons, via the production of neurotrophic factors and by providing metabolites. On top of that, they also regulate the blood-brain barrier and stabilize and regulate the function of synapses. Many of these homeostatic functions are perturbed in the context of inflammation, in the context of CNS diseases. And on top of that, not only astrocytes show an impairment of many of their homeostatic functions, on top of that, they gain disease-promoting activities. And those activities are associated with the recruitment of peripheral immune cells, for example, monocytes via the production of chemokines, the activation of CNS resident cells such as microglia via the production of chemokines and other factors we'll describe later on. And also astrocytes have been shown to have intrinsic neurotoxic activity. Now, when we think about these homeostatic functions that are lost by astrocytes in the context of CNS pathology, and these pathogenic functions that are gained by astrocytes in the context of pathology, it's important to revisit two concepts that were put forward more than 100 years ago by Santiago Ramón y Cajal. The first concept is the idea that actually astrocytes are highly heterogeneous. As you can see here with his simple microscopy methods, he was able to identify different types of astrocytes, at least based on its morphology. So one of the questions we'll try to address through my talk is trying to understand a little bit about the heterogeneity of astrocytes. Second point that Ramón y Cajal made was actually the fact that astrocytes are highly interconnected with other astrocytes and also with other cells within the central nervous system. So with that in mind, first point, what do we know about astrocyte heterogeneity in the CNS? Well, when we think about different astrocyte subsets, right, we can think first of all as those subsets that are established early during development. This is what we call developmentally induced astrocytes. And just to give you a clear example, if we analyze astrocytes from the CNS, from the brain and the spinal cord, we can identify, and there have been reports of very specific transcriptional programs that result in different functions associated with different astrocytes. In terms of, on top of that, even in different layers of the cortex, there are reports of astrocyte heterogeneity. However, on top of that, astrocytes, for example, in the context of pathology, are going to be exposed to multiple stimuli. And these stimuli can actually induce different astrocyte responses or activation states. You can think about these stimuli as cytokines, as pollutants, microbes, or even the accumulation of A-beta plaques. Each one of them can trigger specific astrocyte responses. So at this time in research, it's very hard, and we're really getting at the beginning of trying to understand what of those collections or families of astrocytes we define are actually developmentally induced or stimulus induced. That's why throughout my talk, I'm going to refer to them as astrocyte subsets without a clear implication on whether they are induced in development or in response to specific stimuli. So one of the goals of my lab has been to characterize astrocyte subsets in health and disease. And in order to do so, we actually combine a series of bulk and single cell transcriptomic, proteomic, and epigenetic techniques in order to analyze clinical samples obtained from multiple sclerosis patients and controls, and also samples obtained from animal models of multiple sclerosis. And our goal was, first of all, to identify astrocyte subsets, then to actually identify the transcriptional regulators, right? The molecules, the pathways that regulate each one of these subsets, and then actually to perform what we call perturbation studies, where we inactivate each one of these regulators, particularly using the CRISPR technique, as a way of trying to define the function of each one of these subsets in vivo. So with that in mind, what I want to leave you with is actually two examples of what astrocytes can do. And these are examples that really sit at opposite ends of all the different functions that astrocytes play in the context of CNS pathology. And this will be helpful because then that would provide a framework to understand what I want to talk about in the second part of my talk. So a few years back, we identified a population of astrocytes, which is actually controlled and driven by the transcription factor MAFG. This is actually a population of astrocytes that is significantly expanded in the cortex and the cerebellum of multiple sclerosis patients, and also in animal models of multiple sclerosis. Some interesting aspects about this specific astrocyte subset is that it shows a decreased ability to support the metabolic needs of neurons. And this is something that actually Chun-Che Chao in the lab studied in depth. In addition, this expanded astrocyte subset shows a deregulated unfolded protein response. This is something that Mike Wheeler has studied recently, and there's actually in the lab, and there's actually new work coming there. And that's important because this deregulation of the unfolded protein response might provide novel targets for therapeutic intervention. And the last point I want to make, which is very interesting, is the fact that this pathogenic astrocyte subset, right, if we inactivate it, we can significantly ameliorate CNS pathology in animal models. One very important aspect about this astrocyte subset is that it's actually induced by GM-CSF, a cytokine produced by effector T cells. In other words, once these T cells make it into the CNS, they produce this cytokine GM-CSF, and that actually promotes or boosts the differentiation of these pathogenic MAFG-driven astrocytes. So this gives us an example, is a clear example of a subset of astrocytes that are highly pathogenic. Now, at the other end of the spectrum, we recently characterized an additional astrocyte subset, which is characterized by the expression of this molecule here called TRAIL. TRAIL is a classic molecule many of you, if you have training in immunology, might have heard about it. It's a molecule known to trigger T-cell apoptosis. Indeed, what we found is that these TRAIL-positive astrocytes seem to line the borders of the CNS and basically induce T-cell apoptosis as T-cells try to enter into the CNS. Interestingly enough, this anti-inflammatory astrocyte subset, right, it's actually induced by interferon gamma, which is produced, among other cells, by NK cells circulating through the meninges. So this is actually an important point, and actually brings me to what I want to discuss with you now, which is the fact that, as I mentioned, it's not only that there are different astrocyte subsets, they seem to be highly interconnected with other cells in the CNS, and those connections actually play central roles in astrocyte function and eventually in CNS physiology and pathology. And just to give you an example, we talked briefly about astrocytes and their interactions with T cells and NK cells and how that could regulate pro and anti-inflammatory astrocyte subsets. We have discussed also previously the interactions of astrocytes with neurons and oligodendrocytes. And we and others have touched extensively about interactions between microglia and astrocytes. The question or the challenge, though, is that each of these interactions, right, was identified as the result of very deep studies, but there's no method that allows you to interrogate cell-cell interactions in vivo in an unbiased and comprehensive manner. So this we saw that as an important challenge that was stopping or limiting progress in the field. And in order to address that, what we did is we developed a new technique, which we call Rabbit-seq. And basically, this is a technique that uses the rabies virus as a vector in order to transmit or to transfer a genetic barcode to cells that are interacting in vivo. Why is that important? Because then based on this genetic barcode, we can actually reconstruct cell-cell interactions in vivo, and by doing so, reconstruct or identify potential mechanisms of cell-cell interaction. And this is actually one of the examples of one of the first times where we use this technique. We first use it to study the EIE model of multiple sclerosis. The first outcome you obtain with these studies is actually shown here, and it's kind of similar to what you've seen by now hundreds of times, Disney plots or UMAP plots, where each one of the dots represents a single cell for which you have transcriptional data. However, what is different in this technique, as opposed to other techniques out there, is that each one of those dots is annotated with a barcode that allows us to reconstruct the interactions of that particular cell in vivo. Hence, we can transform a dot map like this one into a network interaction map like the one shown here, where at the core of it, at the base of it, we can identify each one of the interacting cells and also try to identify the specific mechanisms mediating those interactions. Hence, once we construct these networks, we can also reconstruct interactions and how they are altered in health and disease. And we can identify the interacting cells, we can identify the interaction mechanisms, and we can define the downstream effects of those interactions. So, like with any other new technique, the first thing you want to do is to first demonstrate that you can reproduce previous reports. And we were happy to see that, first of all, with using Rabbit-seq, we were able to reproduce previous reports actually coming from my own lab, showing that microglia can control astrocyte responses via BGF, and also reports from the Ben Barres' lab, where which reported that C1q, IL-1, and TNF produced by microglia can control astrocytic responses. Hence, Rabbit-seq can detect interactions between astrocytes and CNS resident cells. We're also happy to see that Rabbit-seq was able to reconstruct interactions between astrocytes and peripheral immune cells recruited to the CNS in the context of inflammation. For example, interactions established between astrocytes and TR1 cells mediated by IL-10. So, in summary, Rabbit-seq is actually detecting previously described interactions. So the question now is, can we use it to learn new biology about astrocyte regulation? In order to do so, we decided to focus on the most pro-inflammatory astrocytes as based in their transcriptional profile. And then based on that, we reconstructed their interaction networks, and we tried to identify those microglia interacting with these highly pro-inflammatory astrocytes. We then identified the molecular mechanisms mediated those interactions between highly pro-inflammatory astrocytes and microglia. And to our surprise, at the top of the mechanisms mediating those interactions were axon guidance pathways. As you know, those are pathways that are very known and very active during development, but which were not previously described to mediate interactions between astrocytes and microglia in the context of CNS pathology. In particular, we detected an interaction between the F receptor and its ligand ephrin. So a little bit of background on the F receptor. The F receptors constitute the largest family of tyrosine kinase receptors, and this is important because that means that here in this intracellular domain, you have a kinase domain, which could potentially be targeted therapeutically. And we'll get back to that. In particular, our studies identified FB3, the F receptor B3, as a candidate molecule expressed in astrocytes. And interestingly enough, we detected as a candidate molecule activating it as its ligand ephrin B3 in microglia. And what's important about it is that in this case, not only the receptor, but also the ligand, ephrin B3, both of them are membrane bound, and both of them have an intracellular signal domain that allows them to trigger signal transduction. Hence, interactions mediated via FB3 and ephrin B3 have the ability, potentially, to modify the responses of both interacting cells. These are actually bidirectional interactions. So based on these findings, right, and in this literature, we decided to test or investigate the role of FB3 and ephrin B3 signaling in CNS inflammation. First thing we did was actually to inactivate FB3 in astrocytes or ephrin B3 in microglia. And as you can see here, but even before getting to that, the first thing we wanted to do was to see whether we could detect the expression of these molecules in clinical samples. And as you can see here, we detected a significant upregulation of FB3 in astrocytes in MS lesions. And we detected a significant upregulation of FMB3 in microglia located in the same MS lesions. Even if we go downstream and we analyze single cell data from those lesions, we also detect a significant upregulation of FMB3 signaling in microglia and FB3 signaling in astrocytes. So now if we perform perturbation studies, basically we inactivated the receptor FP3 in astrocytes or its ligand in microglia, and that results in a significant amelioration of EIE and also a significant decrease in astrocyte pro-inflammatory disease-promoting responses. And this was repeated and recapitulated in many EIE models, in the chronic model, the acute model, so on and so forth. Now, how does that work at the molecular level? Basically, we interrogated what's going on downstream of this signaling domain. And to cut it short, what we found is that FB3 signaling controls mTOR signaling, and by doing so, controls mitochondrial function and dysregulation. And indeed, in new work produced by Atsushi Kadowaki in the lab, we have really got into the molecular mechanisms by which mitochondrial dysregulation astrocytes might play a central role in neurodegeneration. Now, last point I want to make is, as I mentioned earlier, this is actually, since both interacting proteins are membrane bound and both interacting proteins harbor a signaling domain, this interaction is what we call a bidirectional interaction. Not only astrocytes are going to change their responses in response to these interactions, also microglia. And to cut it short, what we found is that when you inactivate this signaling pathway in microglia, you see a significant decrease in NF-kappa B signaling. So the last point we wanted to make is this is a very interesting pathway. From a basic point of view, is there any hope or any tool or strategy to target it therapeutically? In order to address that point, we actually screen several libraries, and in particular, we screened a library of small molecules, which because of their chemical properties were known to be CNS permeable. And to cut a long story short, we identified one molecule, which we call A38, which is a strong inhibitor of the intracellular tyrosine kinase domain of the FP3 receptor. Indeed, if we actually administer this molecule to EIE mice using either the B6 model or the NOD-EIE model, that has the ability to strongly suppress EIE development, and in particular, strongly suppress pro-inflammatory responses in astrocytes and microglia. So to summarize, first of all, we developed this new technique, which we call Rabbit-seq, which allows us for the first time to study in a comprehensive and unbiased manner cell-cell interactions in vivo and the mechanisms that mediate them. We actually recently extended this technique, and we have improved this technique so we can study clinical samples. And for example, we have recently used it to identify mechanisms driving immunosuppression in the tumor microenvironment of GBM patients. We have now extended this new technique to the study not only of glioblastoma, but also of clinical samples from multiple sclerosis, living monkeys in the model of aging, and even for the study of neuroimmune interactions in the gut. Second point I want to make is that actually we were able to identify novel mechanisms, unknown mechanisms mediating astrocyte-microglia regulation and dysregulation in the context of pathology. And in particular, that led us to identify one molecule, A38, which we think might be a base compound, right? A lead compound in order to develop small molecules of therapeutic use. And with that, I'm going to finish. I just want to thank the organizers for giving us the chance to present our data and acknowledge the work of Mike Wheeler, Cristina Gutierrez Vasquez, Ian Clark, and Liliana San Marco, who are the scientists that drove this project.
Francisco, very beautiful presentation. I have a question for you that many people, you probably haven't been asked. Are there differences in astrocytes between different species, like the mice or the people, or that we're studying? I mean, go ahead. Yeah.
That's an excellent question. So it is believed that actually human astrocytes are more complex, for example, than rodent astrocytes. And indeed, there are some studies that with the humanized mice that suggest that mice harboring human astrocytes might have some improvement in, let's say, cognitive function.
Right. And another question, is there a way to measure or see these different subsets of astrocytes in people, either through imaging or proteins in the spinal fluid or blood? Is there any, I know there's studies on NFL which relate to neuronal damage. What are your comments on astrocytes and specifically say GFAP, which we're measuring in the blood?
So those are excellent questions. First of all, some of the PET markers we use, we now know that they have some confounding noise or signal coming from astrocytes. Those are markers that we use to PET probes that we used to think were specific for microglia. So that is actually important because some groups are actually working on developing astrocyte-specific probes for PET. On top of that, as you suggested, some investigators have been working on the idea of measuring GFAP and other astrocyte markers as potential biomarkers for neurodegenerative diseases, and those results seem promising, though they haven't been as thoroughly validated as NFL.
Very good. Again, thank you very much. Beautiful work in a very exciting area. We're going to continue with our symposium now on innate immunity in the central nervous system. The other major cell in the brain, of course, is the microglial cell. And I'd like to invite Oleg Butovsky to speak to us about his work and on microglial cells in the brain. Oleg?
Thank you, Albert. I'm going to share my screen. Let me know if you can see my screen, okay? Can you see my screen? I guess so. I didn't hear yes, no, but I guess okay. All right, so Howard, thank you. And I would like to thank the organizer for this small, although important forum symposium. As you know, microglia are the major innate immune cells, I would say, in the brain. They are almost first responder to any disturbance to the CNS. And I would like to speak today on actually an opposite role of microglia in neurodegeneration. And I will shed light on really new directions we have in the lab. So let me start. Several years ago, my lab and others identified two major microglia subset. The first one on the left side is called homeostatic microglia, and on the right side is called microglia neurodegenerative phenotype, or other people call it disease-associated microglia. Now, the most importantly, this became available, identification of the signatures and the role and functions because of the technological revolution in the field. And secondly, and the most important, I think, the mechanism, how the cells behave, regulated, that was as well, have been identified partially, although. But what we know today in terms of homeostatic regulation, the TGF-beta tumor growth factor beta signaling is the driver, is probably the most important upstream regulator to make or maintain microglia in normal, healthy, adult homeostatic environment in the brain. Now, once microglia exposed to any perturbation and specifically any injuries or cell death, this particular mechanism involved, which we identified, we called ApoE-TREM2 signaling, and via this mechanism, microglia sense apoptotic cells, neurons, or any other cell types, and they immediately, within hours, switch to this neurodegenerative phenotype, which can be read by molecular signature, specific markers, and so on, etc. One of the really biggest questions is whether these two phenotypes are they truly bad or good? And this is really standing questions for many decades. And these questions should be really addressed to know how to approach, how to treat disease, how to read disease, how to read, say, biomarker of microglia's respond to type of treatment in clinical trials. So, and I'm using this Twitter from Ruslan Medjidov, who is really a great scientist in innate immunity, actually. And he proposed this schematic where he sees cells get inflammation, and they can get out from homeostasis to inflammation. But once the cells get to chronic inflammation, they seems to be stuck. And the biggest question, can we reverse their fate, their stage to back to homeostasis? Is it good or bad? So this is the type of question I'm going to touch. I'm going to use this beautiful cartoon which we published on Immunity cover paper, symbolizing bad and evil or good, or in this scenario is Dr. Jekyll, Mr. Hyde. And let me start. So a few words about homeostatic microglia, what they are. Again, several years ago we described, identified this molecular signature, and the most importantly, the mechanism of regulation, which is regulated by TGF-beta signaling. But interestingly, one of the molecules which came to our attention, although we didn't pay attention, it was supplementary data, it's called ApoE, apolipoprotein E. And this analysis, this ingenuity pathway analysis, actually predicts or says, if you want to have the microglia homeostasis regulated by this signaling, the ApoE cannot coexist. It's obviously going to be in opposite way, so it's in blue, it's down. Okay, I'll get touch about this. And now the question why and what it does. Now, as I mentioned, this was really a technological revolution, not to mention just Frank showed. It's really amazing technology what he's developing. Single cells, all this spatial technology. But obviously the basic one is to develop these microglial specific markers, so antibodies, which we did in my lab in Harvard when I was postdoc. So here's just an example of these molecules. And then in order really to validate, confirm what this TGF-beta signaling is truly the upstream or the master regulator, we developed this very interesting mouse model which do not develop or not express only in the CNS TGF-beta ligand signaling, okay? And what's happening in these mice, either in early stages, postnatal stages, or later on, they do not express any microglial homeostatic molecules. Again, I emphasize homeostatic molecules. But interesting, there's another one called EBA1, which is very widely used. It's a pan-myeloid marker, always as well used to label microglia, but although it's not specific. So you can see what's still in this complete knockout or CNS-deprived TGF-beta mice in the brain. The cells still exist, but really not resemble microglial morphology. And I put this again cartoon to symbolize this Mr. Hyde, this evil guy who eating the cells he was drinking by the novel, by the book. He was drinking serum to become evil guy. But you can see all the cells are really attaching and kissing and really seems to be swallowing neurons in the brain once they don't have TGF-beta, okay? Now, we did all this profile, and one of the major molecules which induced in these myeloid cells, once they don't have TGF-beta, was ApoE. It was skyrocketing, thousands-fold induced, and I'll get to this just in a moment. Now, the time these mice developed very interesting phenotype, they look like they have some neurodegenerative or some paralysis-related disease, all these symptoms you can see. This is two siblings, two brothers. One is TGF-beta CNS deprived, one is normal mouse. But this mice, believe me or not, is going to be completely paralyzed in about 6 months of age, and it's going to be 100% lethal. So the loss of TGF-beta signaling in the brain lead to this severe phenotype in the mice. But again, and as I mentioned, TGF-beta is a primary, seems to be regulator of homeostatic microglia. Later on, others, different labs came to the same conclusion using different approach. This is an example from Chris Glass' lab, David Gozelin, who did epigenetic studies, and he came to the same conclusion. What PU1 in cooperation with SMAD signaling, particularly SMAD3, maintain microglia homeostasis. Now, later on, the tools became available, and now we can specifically manipulate only in microglia, so we can deplete or delete TGF-beta signaling conditionally, so we don't have to, to have this problem from beginning, from embryogenesis, but now conditionally we can kill TGF-beta signaling microglia. And what we found was just exactly the same phenomena, same results. The microglia lose their homeostatic signatures delineated here by P12 signaling, and the mice again develop severe paralysis and death. But is not just loss of physical loss of microglia. What's happening, the microglia transitioning, once they don't have TGF-beta in brain milieu, they transition to the novel phenotype, which we call MGND, microglia neurodegenerative phenotype. And again, you can see these classical molecules, again, as an example, ApoE was, again, thousands-fold induced. It's not probably, is the most induced out of 1,000 genes in microglia once they transform to be MGND. And this is upon, again, losing TGF-beta signaling. And our colleague, again, here from Children's Hospital, Team Springing Lab, published, again, using similar approach, different approach by manipulating the TGF-beta signal. In this scenario was LRC33, which is co-adapter of TGF-beta activation. He found exactly the same results: loss of homeostatic microglia associated with demyelination, paralysis, and death. Now let me move forward and define the MGND microglia and the mechanism of regulation. So these two amazing previous postdocs in the lab, Susan and Charlotte, now became professors in Bordeaux, France, and Hamburg in Germany. They took really a big effort to, first of all, identify commonality of microglial mechanism of regulation, how they look like, those barcodes, in microglia and several disease mouse model, and obviously validation in human AD brains and other major neurodegenerative diseases. And the signature have been identified, which again called today MGND, or Idomit Lab, who published similar story about DAM, disease-associated microglia. And again, you can see that I'm just showing you two major clusters, but ApoE was among the most induced genes, and you can see that disease progression by score is associated with suppression of these top microglial transcription homeostatic regulators and reciprocal induction of ApoE as disease progressed. And just to summarize this interaction, again, I just want to emphasize these two molecules cannot coexist whenever we have suppression of TGF-beta signaling in neurodegeneration, specifically in microglia. ApoE will be induced. And later on, we found what ApoE basically plays as a major regulator to make this transition, which I will just show in a moment. But beforehand, this gives us an opportunity to develop novel tools. And now we introduce this CLEC7 molecule, which specifically can only label MGND microglia associated with cellular damage. In this scenario, this is plaques in AD brains, mouse, and human. Next slide. And you can see this red color, the microglia, once they get close and in association, physical association with the neuritic plaque. I want to emphasize neuritic, which is associated with cellular axonal damage and apoptosis. The microglia became MGND. So this picture shows transition of this homeostatic towards MGND. Now we developed novel mouse model where we can specifically, is based on CLEC7 CREART2, which can specifically and conditionally manipulate with only microglial subset associated with neurodegeneration. It's quite important. Our colleague and friend Hans Lassmann validated this observation in human AD brains. And again, this is just an example of diffuse plaques, so-called, versus neuritic plaques in AD brains. And again, diffuse plaques are not yet associated with apoptosis or cellular damage. It's just segregation of the plaque. It's in early stages. But later on, these plaques will kill or be cytotoxic and will be associated with neuritic plaques and basically cell damage. And you can see what microglia completely lost their homeostatic identity. Although they exist there, but we cannot see them anymore. So they became MGND. And just to summarize the cartoon of these dual faces of Dr. Jekyll microglia degenerating neurons cannot hide with Y, but it's truly whenever case microglia sense apoptotic cells, it doesn't matter, neuron astrocytes or leukocytes or any immune cells, they have machinery such as ApoE2 to sense the apoptotic cells and became MGND phenotype. Now I show here by shadow this evil guy, but I'm probably going to convince you not always this bad guy is truly bad guy. So in order to study the functionality of these cells in vivo, we introduced this MGND paradigm by injection of apoptotic neurons into living mouse brain. And within us, you can see what microglia delineate the percent of injection, and truly within 16 hours, they transition to MGND phenotype as shown here on heat map by this gene expression profile. Again, ApoE was the top induced molecule, whereas TGF-beta signaling, among others, homeostatic regulators and molecules were suppressed. In order to address the role of ApoE and the question what ApoE does, why it's the most induced gene in microglia, until today we know the major source is astrocytes. In order to address this question, we implemented this MGND paradigm by injection of cells into wild-type brain versus a global ApoE knockout mice, and we found that it was pretty interesting. It was sufficient to have cell-increasing effect of ApoE in microglia only, meaning if we remove ApoE from microglia only but keep everything else, such as astrocytic ApoE, we're going to get the same results. The microglia will not be transitioned to MGND phenotype. So it's truly show cell autonomous regulation of ApoE in microglia. It's quite interesting. Now the next question was, what ApoE does to microglia transition? Is it good or bad? Is this MGND good or bad? This is the first case we did, and this is fascial exotomy, where we keep brain intact, but we injure the peripheral nerve, which retrospectively or retrogradely induce loss of fascial nucleus, as shown here. This is wild-type mice, and this is injured mice. You can see complete loss of fascial nucleus. There is deletion of ApoE specifically in the microglia. Global deletion actually preserve or somehow neuroprotective the neurons. So it shows you what this phenotype, which is called MGND ApoE mediated, is basically cytotoxic in this model. Later on, in collaboration with Dave Holtzman, we did this work where we used tau mice as tauopathy. This is a classical neurodegeneration mouse model. You can appreciate this lesion site. But the most exciting to me was what showing that global deletion of ApoE in this traumatic mouse model completely protected loss of neurons. Although this was global knockout, so it was not clear exactly who contributed what. And this is Caroline from my lab participated in this study and found what global deletion of ApoE in this mice as well preserved microglia at homeostatic stage. So that's kind of showing us what, well, it is possible that actually ApoE drives, again, cytotoxic phenotype. I'm going to switch a couple of minutes of my last talk about the major risk factor, which is called ApoE4. And again, microglia has so many different regulators and interesting risk factors, particularly in Alzheimer's, about 170. But one of them is ApoE4. It's truly the major risk factor. However, ApoE4 is associated with a reduced risk of glaucoma. This was interesting once we learned that and say, well, I think we should look in parallel both diseases, at least mouse model, to understand what is the mechanism. So let me show you, okay? Now, as I mentioned, there's a big consortium we did, who led by my previous postdoc. She became now professor at Harvard, Massanir, Milica Margetta. This is the biggest JIVA study, and again, it shows that ApoE4 is protective for glaucoma. Now, to address this question, we use two major mouse models of glaucoma. The one is called microbead injection, which basically clog blood vessels in the eye, and by this inducing internal pressure in the eye, which leading to loss of retinal ganglion cells. The second is called DBA2J mice. This is based on genetics, and these mice as well develop loss of retinal ganglion cells. Both models show more or less similar results. Among them was ApoE as the most induced molecule in retinal microglia. Whatever from now on, I'm going to talk about microglia in the eye. Okay. Now, interestingly, to address direct effect of microglia on RGC loss, what we did it was very interesting experiment. We transplanted from brain MGND microglia, which induced by apoptotic cells, directly into the eye. Now, this gives us an opportunity to manipulate of microglia in this brain, including deletion of ApoE. So, and what we found just by using transplanting the cells, MGND phenotype for wild-type mice versus ApoE knockout mice, we found complete block of loss of RGC of microglia which do not express ApoE. So again, this model shows that ApoE truly drives microglial phenotype to be cytotoxic. In terms of functionality as well, I'm not going to go through details, but as I mentioned, ApoE4 was the most protective allele. So we confirm now using humanized ApoE2, 3, and 4. In human, we have three major ApoE variants. and 2 is the most protective allele in Alzheimer's, okay, it's opposite for glaucoma. But we confirmed what in ApoE4 microbits injection mice, they are mostly protected, okay, as shown here, this is retinal ganglion cell, they're less lost as an opposite to 2 or 3 alleles, okay. Now, in terms of molecular regulation, we're finding very interestingly that in ApoE4 microglia, this microglia cannot, I'm sorry, my screen's jumping. The microglia from the retinal ApoE4 mice cannot induce the MGMD signature as this happening in microglia expressing ApoE3. This was something very unique, and again, supporting the idea what MGMD phenotype is cytotoxic, but it shows something very interesting or something novel, what ApoE4 somehow blocks or interfere the microglia to induce the MGMD phenotype in the eye, excuse me. We confirmed this as well in human glaucoma eyes. This is human tissue, and we found that one of the molecule called galectin-3, which is a part of MGMD signature, is only induced in ApoE3 donors, glaucoma eyes, but not in ApoE4. So again, validation in human specimen that MGMD microglia is affected, it's impaired to induce this phenotype in ApoE4 donors, okay? To summarize this part, what we think happening is the following. I'm not sure all mechanism, but galectin-3, this molecule, was the primary molecule which is cytotoxic to retinal ganglion cells, which is induced by ApoE3, which is the primary regulator of microglia to be converted from homeostatic to MGMD phenotype, and this way microglia express high level of galectin-3 and directly kills RGCs, retinal ganglion cells. There is in ApoE4 donors, microglia are impaired to induce MGMD phenotype. They stay in homeostasis and they cannot kill RGCs. And then the last question I would like to show, so how come ApoE4 is good for the eye but bad for the brain? So this took us 6 years to address this question, and finally this paper now submitted. And this work has been led by Juran Yin, Charlotte Modor, and Netta Rosenzweig in my lab. And we took advantage of novel technology and models to understand ApoE4 regulation in AD mouse or mouse models. But first what we found, what MGMD microglia, as I just showed you in the eyes, they impaired to induce this MGMD or DAM signature in MGMD paradigm. So again, there's something happening, they stuck in homeostasis. It's just a small part, the thousands of genes are affected like that. This is just a small part of genes related to phagocytic function or antigen presentation. And you can see all these genes are not induced in ApoE4 expressing microglia. This is just naive brains. Now, in the same injection site of dead neurons, microglia cannot accumulate in ApoE4 and express these molecules such as galectin-3. So again, showing you what their response as well to neurodegeneration is limited, okay? The most important, now we can manipulate the expression of ApoE variants in microglia, and we use this novel mouse model where we can conditionally delete human variants in mice, okay? And again, you can see here, just very briefly, you can see what deletion of ApoE4 in MGMD paradigm, again, response to dead neurons, restore their response. So this is something very interesting because it shows it's not loss of function, it's a negative regulation of ApoE4, and by deletion of ApoE4 in microglia, we can restore their response to neurodegeneration. Next, we tested whether how ApoE4 plays a role in microglia, specifically in AD mice, and in this scenario, this is tauopathy, P301S mice. This is the mice which develop very severe neuronal loss. And remember, I mentioned before, there's one of the markers we discovered and described is called CLEC7A, which specifically stain MGMD subset, the microglia which respond to neurodegeneration, right? And you can see truly like black and white signature just based on CLEC7 expression between ApoE3 knock-in mice, microglia in tau mice, they have robust response that became MGND based on CLEC7 and the molecular signature. However, the ApoE4 knock-in mice do not express or very limited an expression of the signature. But again, once we conditionally remove ApoE4 in microglia mice, we restore their response to neurodegeneration. That was the most importantly associative protection. So this is the first result showing that by manipulation of specific genes such as ApoE4 in this scenario, will release or restore microglial response to neurodegeneration, which we thought they are cytotoxic. They express multiple genes associated with cytotoxicity inflammation. But in fact, this was associated with neuroprotection. The next model we tested was Alzheimer mice. This is APP-PS1 mice. And we found basically very similar impaired response of microglia. And again, removal of ApoE in microglia specifically restored their response. But now, not only that, we found that microglia, once they became MGND, they start to talk to astrocytes. And Dr. Quintana just beautifully showed the dialogue, on several levels, mechanism, how microglia astrocytes can crosstalk each other. So we found that by restoring MGND phenotype, I'm going to show next slide, microglia not just became CLEC7 or MGND, they start to call upon astrocytes to come to the plaque and encapsulate the plaque. Again, this was associated with reduction in plaque load, which is one of the major manifestations of Alzheimer's disease progression. So here's an example of Alzheimer mice expressing ApoE4, okay? The white cells are microglia surrounding the plaques. But once we remove specifically in microglia ApoE4, astrocytes became activated and encapsulated the plaques. And this is another example using different markers of activation of astrocytes called serpina-3N. And we basically find the same phenomena, same results, reduction in the plaque and encapsulation of astrocytes. Again, this was done via microglial manipulation, and we see now how astrocytes responded. So basically, there's direct dialogue between microglia and astrocytes, and I'm not showing all details, but we identified exactly the mechanism how this is happening. Now, again, to address the direct role of microglia with different variants, so again, we're using this microglial transplantation experiment. Now, in this scenario, donors are 3 knock-in versus 4 knock-in versus 4 knockout microglia. We transplant them into naive brains and look what's happened to astrocytes. And we found that, deletion of ApoE4 in microglia not just restored their phenotype, but it's induced activation of astrocytes. So again, the MGND phenotype, which is impaired by ApoE4 and restored once deletion of ApoE4, talk to astrocytes, activate astrocytes, which seems to be beneficial for Alzheimer's. Now, the last few slides, I'm going to show you what's happened in human brains, and this is what's important to validate our findings. So in this scenario, we used human donors, 80 brains expressing different variants, such as 3-3 or also 3-4. It's almost impossible to find homozygous for, so most of the brains are 3-4. And we found a big deal with sex differences, and particularly in females expressing 4 variant. We found what in their brains, there's much more induction of TGF-beta signaling, such as denoted by SMAD3 here, and obviously suppression of all these markers associated with microglia and astrocytes activation. This is whole brain RNA sequencing, okay? Again, confirming what we basically found in mouse studies. The most importantly, independent validation from different databases, this work by Marco Colonna lab, Zhu et al., using now single nuke-seq analysis of microglia, basically we found the same activation of TGF-beta signaling in microglia expressing ApoE4 variants. So again, it's very important to show all this confirmation. And remember, this galectin-3, I mentioned several times, was suppressed, such as in glaucoma as well, in 80 brains, and we confirmed that in human 80 brains. Here's ApoE3 80 brains. The red is galectin-3 specific expressed in microglia. We cannot see this in ApoE4 donors. And this activation and expression of galectin-3 was associated with activation of astrocytes in 3 donors, but not in ApoE3/4 donors. Again, showing there is impaired crosstalk between microglia and astrocytes to deal with at least with the plaques. Okay. One of the interesting discoveries we made was associated with induction of SHIP1, which is one itself, it's called NPP5D, it's the name of the gene, which itself as well AD risk factor. And we found this was absolutely highly induced in mouse and AD brains expressing ApoE4 variant. Now, just of note, SHIP1 and many others checkpoint, so-called checkpoint regulators of microglia, are regulated by TGF-beta signaling upstream. So if we have brain milieu which is enriched with TGF-beta signaling, we're not surprised to see all these checkpoints, microglial MGMD inhibitors, are highly expressed in AD brains, expressing ApoE4. And genetic deletion of SHIP1s now specifically in microglia restored or enhanced the MGMD phenotype associated with reduction of A-beta loads, okay? So to summarize, and again, here's showing as well, again, deletion of SHIP1 and activation of MGMD microglia activates astrocytes and encapsulating the plaques as an opposite to just, wild-type mice. I have probably my screen. Let me summarize, okay? So what we find with ApoE4 impairs microglia response to acute and chronic neurodegeneration. ApoE4 microglia accumulate lipids and represent dysfunctional metabolic state, so not just what Dr. Quintana showed before in astrocyte, but is very known now what is the lipid microglial dysfunction in most of the neurodegenerative diseases, and ApoE4 drive this. Deletion of ApoE4 in microglia facilitates MGMD response in AD mouse models. Deletion of ApoE4 in microglia decrease A-beta plaque load and LAMP1 dystrophic neuroids in the same mice. And deletion of ApoE4 in microglia promotes recruitment of astrocytes toward plaque associated with reduced A-beta plaque pathology. The most importantly, AD patients carrying ApoE4 allele show repressed MGMD response and astrocytes activation gene expression profile. These data support that MGMD DAM microglia are beneficial in Alzheimer's, okay? And again, I think this is extremely important to create new drug or target development towards microglia, polarizing them towards MGMD phenotype. This is really the final slide I want to show. This is the JAST paper published, again, the cover page. So show dramatic view now of Dr. Jekyll, Mr. Hyde. In this case, ApoE3 microglia are fighter. You see the sword and blood is, as called weak ApoE4 spills down in glaucoma. Of note in Hebrew, dam is blood. But this is in glaucoma. You can see there's inflammation, damage. There's ApoE4 microglia is mediating sleeping, it doesn't care, and there's no death. This situation is quite different in Alzheimer's brain. So we should be careful by studying disease and what treatment to do to particular disease or stages of disease. I'm going to stop. This is my last slide to thank to all my group. And I mentioned a couple of names, but without them, obviously, this work now been done, my sources and all my collaborators. Thank you.
Oleg, beautiful presentation, really interesting work. I have one question for you before we go on. You were the person that created the idea of MGMD, and you had this beautiful picture of Dr. Jekyll and Mr. Hyde. Now you suggest that MGMD may be beneficial, which is very interesting. Do you think there's going to be another classification of these microglia? They'll have a different name than just MGMD. Maybe it's MGMD 1 or 2 or 3. What are your thoughts?
Yeah, of course. We're struggling, I said we as a field of microglia, and particularly neuroinflammation, we're struggling with the definition. And I hope one day we're going to get to T-cell biology field, where T-cell is called by so many names. And it's not just, it's very important because we understand once we call different names, we understand what they do, their functions. So we're trying to do that. I truly believe MGMD will be different subset, we call it differently. The microglia are extremely dynamic. Even within the same disease, as disease progressing, they change their phenotype. So I'm not talking about those small nuances, although very small nuances are very super important as a target. Okay, so yes, it's very important to come with more defined names which will represent or show what they do, the functionality. And we really like, we're not in advanced mode as a T-cell biologist, to understand what they do. So there's a lot of to be done yet.
I'm going to ask you the same question I asked Fran. Is there a difference in microglia between human and mice? I mean, we study both.
Yes, of course. Well, yes, and I say yes, the sadness, because one of the biggest limitations is to study human brain in life. So all this data we see today, and it's extremely hard to deal with human brains as to study the microglia astrocytes. It's a postmortem tissue, and the cells are quite dramatically changed, particularly using nuke-seq rather than whole live cell RNA sequencing. And we do see, actually comparing two data sets from Marco Colonna, who use nuke-seq, and for example, Marta Oleg, our previous colleague, Phil Dejagi lab, they use live, fresh postmortem tissue within hours. You can see dramatic differences, not just in the level of depth of the analysis, but as well the quality. So my answer would be probably they are different, Howard, but I cannot jump to say how much are different because of technical limitation to study human versus rodents, okay?
Okay, Oleg, thank you very much. Obviously, the microglial cell is one of the key innate immune cells in the brain, and the field is actively evolving. Again, thank you very much. We're now going to the third talk by Rafael Rezende. And now we're going to begin to look at ways to modulate innate immunity in the brain with therapy. And Rafa is going to talk on modulation using modulation of microglia, which Oleg just talked about, with nasal anti-CD3. So Rafa, please begin.
Okay, thank you, Howard. Thank you everyone for the invitation. As Howard mentioned, we're going to talk about, I've tried to summarize over 30-year work by Howard in 30 minutes, which is not going to be easy. But the title of the presentation is How Can You Modulate Microglia with Nasal Anti-CD3 for the Treatment of Neurodegenerative Disease? So I won't get into much detail on the microglia biology, as Oleg beautifully described them now. But I'm just going to put this figure as an overview of what happens in several neurodegenerative disease. For example, multiple sclerosis, TBI, or AD, stroke, in which several cells play a role in the inflammation, like Fran told also beautifully about these astrocytes. But the microglia, it's as an innate immune cell in the brain, play an important role in producing factors, cytokines that promote neuroinflammation. And as Oleg mentioned, there's these two types of microglia so far that we understand, which is the homeostatic microglia and the neurodegenerative microglia, MGND or DEM. And they play different roles. And also Oleg showed like what Rosalind mentioned, which is like there is a homeostatic function, and when there is an insult, there is an optimal activation that needs to happen to, help the tissue to heal or to clear. But then for some reason, things go on the other way towards neurodegeneration. It's an inflammation that becomes chronic. And we need to find ways to make it go back to this optimal activation or homeostasis and make the disease better. So that's the biggest challenge for everyone in the field now. And then the question is, and how can you modulate microglia activation? So there's several ways it can work centrally that people are being investigated. But as in our lab as a T-cell lab, so we thought about some roles for peripheral immune cells that could, regulatory cells that could go to the brain and interact with microglia or producing some cytokines, regulatory cytokines that would suppress the inflammation. So, and then one of the biggest systems that Howard started work, like as I mentioned, over 30 years ago, is the induction of Tregs by stimulating the mucosal immune system. And he started by investigating the oral induction through oral feeding of antigens to some disease to induce regulatory T cells against that antigen, which would control the disease. We call that oral tolerance. This is a physiologic and safe mechanism. And he published several papers regarding the role of oral tolerance in disease, for example, EAE, which is a multiple sclerosis rodent model. And again, it keeps showing that even in the double blind pilot trial of oral tolerance worked in people as well. And there is other ways, like even for other autoimmune disease, such as rheumatoid arthritis or lupus and other disease, they work beautifully in animals and at some extent in people. But a phase 3 trial actually failed. And then there was a need to find out novel ways to manipulate these T cells in the periphery and eventually help them, to control neurodegenerative disease. And an alternative method to induce Tregs would be oral or nasal, as I'm going to talk a bit later, but the anti-CD3. So the idea Howard had, and this is his own words, and he came to the lab one day and said, what if you give orally anti-CD3? That's a crazy idea. So Howard has lots of these crazy ideas that actually work. And this is one of them that it's really amazing. The point here is that we use antibody to bind the CD3 co-receptor. It's a co-receptor that, as you know, works together with the TCR on a T cell to induce their activation. So there's a big difference if you inject anti-CD3 intravenously, which would lead to a massive activation of T cells and eventually death of these T cells. But what about mucosal administration of CD3, anti-CD3? This hadn't been tested yet. And what one of the first experiments that was done in the lab was giving feed this anti-CD3 in different models of EAE in different doses, and see whether the severity of the disease or the clinical score would improve. And as you can see here, in 3 doses, 5, 50, and 500 micrograms fed orally to mice improved significantly the clinical score. And the best dose here was 5 that eventually we, in our test here, we increased to 10. But between 5 and 10 micrograms for 5 days worked in this case. This paper was published in Nature Medicine in 2006. And as I mentioned, it's a big difference from IV injection of anti-CD3 because oral administration does not change CD3 expression, what actually happens in the IV. So, but the problem is for oral, in order for oral tolerance to be induced, it is necessary that the gut is not inflamed, or at least not very inflamed. And now we know that several neurodegenerative diseases also influence the gut. So in many diseases that affect the brain, the gut is also inflamed. So then it comes to the second idea Howard had. What about giving to another mucosal route, the nasal mucosa? So in that case, we began to investigate nasal administration of anti-CD3 in an animal model of progressive MS. And that was a paper that Fran actually mentioned in his talk about Lior Mayo, who was a postdoc in Howard's lab. And he found that by giving nasally anti-CD3, here's where he started treating these animals 3 times a week. And throughout the process, we can clearly see that the clinical score was reduced and oral anti-CD3 was not effective. And that was very interesting because we now know that in some cases, especially with the microbiota world, now that you understand better, that changed a lot, the gut can be inflamed. And in this particular case, which is a progressive model of EAE, this is induced in NOD mice immunized with MOG. So you have this first relapse, and then it becomes like a relapse remitting disease, and then it gets to chronic secondary progressive, as we see in patients. In this case, the intestine is inflamed, as we already know, and the oral anti-CD3 did not work. So, and nasal work beautifully. So in that case, he also showed that by giving nasal anti-CD3, 5 hours later, the antibody was localized in the cervical lymph nodes. So the mechanism began to unfold in this case that the anti-CD3 would work in the cervical lymph nodes and perhaps inducing regulatory T cells. And in fact, we also found in that paper, they also found that there was a modulation of microglia cells, as you can see here. So it was more like a homeostatic phenotype that promotes repair, and also modulation of astrocytes by reducing some inflammatory markers, as you can see here. But the point here is that he found that the mechanism was through IL-10. So if you block IL-10 receptor by using an antibody, you completely reverse or abolish the beneficial effect of the nasal anti-CD3. So the idea is that when you give nasal anti-CD3, it goes to the cervical lymph node where it modulates or induces IL-10 producing regulatory T cells. And these T cells most likely migrate to the brain and modulate microglia and astrocytes. And we are now trying to understand how this interaction occurs. And when you give orally, it's a different way. It's through TGF-beta producing Tregs, which will also work, but the gut needs to be homeostatic, not inflamed. So, and then it went to a point where it was critical to translate this nasal anti-CD3 to humans. And in this sense, Howard teamed up with Tiziana Life Science, which had acquired this fully humanized monoclonal antibody, which is named furelumab. Was a big advance in the field. It seems like the 80s where we had this OKT3 murumab, which was a fully mouse anti-CD3, until now that we have a fully human anti-CD3 called furelumab. So now we began to test this furelumab in people. And in 2019, we tested in some healthy subjects as a phase 1 clinical trial. 3 different doses, 10 micrograms a day for 5 days, 50 micrograms or 250 micrograms for 5 days. And here's like 6 subjects per group, and placebo, 9 subjects. And in summary, what I found is that there was no side effects, so it was a very safe treatment. And we saw some, the best or the optimal immune effects we saw was in the dose of 50 micrograms. And those immune effects include downregulation of genes involved in survival for CD8 T cells, inhibitory induction of inhibitory or regulatory genes in memory CD4 T cells, and several other effects in Tregs and also in monocytes. We found this activation. So it's like a global activation that starts on CD3 T cells that, you know, somehow it is spread out to other cells as well. And again, this is something we're trying to understand. And then we began to thinking of treating human disease with nasal furelumab. And this is a picture of Howard being treated, or actually it's just how we treated. He wasn't treated with anti-CD3, but this is Dr. Tanuja Chitnis, and how this antibody is it's given. It's a very simple and safe way as well. As you guys can see, it's a simple syringe, and there is an adapter here that you can spray this antibody in the nostril. And it was interesting because during this process, we were testing this phase 1 clinical trial. It came the pandemic. And one of the ideas, as we mentioned, I just showed you that anti-CD3 administered nasally actually induce IL-10 producing T cells, regulatory T cells. And we know that COVID it's a disease that leads to the cytokine storm. It's several inflammatory cytokines in the lung, and that cause all the problems and death. And we actually, you know, embarked on this journey doing a trial in Brazil. And I'm not going to show everything, but the conclusion we had is that the nasal administration of anti-CD3 reduce lung inflammation and blood inflammatory biomarkers in mild to moderate COVID patients. And this is just an example of how the lungs looked. So the patients were treated for 10 days. Here's like before treatment. This is control or placebo. And this is the furelumab. And here you see that normal usual lesions, lung lesions in COVID patients, and 13 days later in control. And here pretty much don't see these lesions anymore after 10 days of anti-CD3 nasal treatment. And then Howard and Tanuja and Tarun, they also started, you know, working on the secondary progressive MS here in the hospital. And one of the crucial points here is the microglial PET scan. So if, one of the things that in our animal studies we found, is I just showed you as well, that anti-CD3 is able to modulate microglia and astrocytes, but primarily microglia. For some reason, when you induce those Tregs, the Tregs migrate to the brain and control this microglial phenotype. And we know, as I'm going to show you, that in the MS patients and other neurodegenerative disease patients, they have activated microglia. So in the treatment cycle here for one patient, actually started, I have the data here from 2 patients in MS, progressive MS. So the nasal pherelimab, the 50 micrograms per day, 3 times per week for 2 weeks, and then followed by 1 week without treatment. And then, the first dose started last year in May, and then it stopped. And they have all the cycles here, and 6-month dosing period that ended on February of this year, 2022, and then it restarted again on March. So it was additional 5 cycles. And what we found was pretty much like a stable EDSS and even improved in pyramidal function, but especially in the timed 25-foot walk, as you can clearly see, the patient improved its ability to walk. And one thing that I mentioned about microglia, so Tarun Singhal, he's developing this imaging and using this PBR06, which binds to TSPO, primarily expressed in microglia. So it's a PET ligand receptor, and for this PET, for this ligand, and now you're able to measure, to image microglia, and you can see here. This is a pre-nasal pherelimab treatment in that patient, and primarily you see an activation of microglia in the thalamus. And here's a different views of the brain. And here's after 3 months, like a striking reduction of activation, which was maintained even after 6 months of pherelimab treatment. And what is important that this thalamic activation correlates with EDSS and time 25-foot walk. So it all makes sense. Here is in several brain areas that was measured. You can clearly see the path in all areas like whole brain, cortex, thalamus, white matter, and cerebellum. They're all going down. So which means that anti-CD3 is indeed reducing microglial activation and improving the symptoms. And then we got an expanded access to patient number 2, a 42-year-old male, was diagnosed with secondary progressive MS since 2014. So, and then again, the first dose were in January this year. There's no safety issues, and 3 months PET in May 2022. So this is what you see at a baseline, very similar to the first patient. And here, 3 months after anti-CD3 treatment, you also can see the reduction in microglia activation in several brain areas. And and this is like the clinical improvement observed at 3 months: increased strength in muscle, improved sensation of the left leg, foot, and faster walking speed. And now the FDA allowed the treatment of 8 additional non-active secondary progressive MS patients with nasal pherelimab. And I think this is starting this month or next month. And then we go to test in several other diseases. But what I'm showing here, the idea is to go next to patients as well for Alzheimer's disease, which you know that is a neurodegenerative disease characterized by accumulation of extracellular amyloid plaques, neurofibrillary tangles, and also microglia activation. So even in humans, you can see that in AD patients, the microglia is very activated compared to healthy controls. And this microglia activation, the tau and A beta, they are also correlated with this microglia activation. So it's a perfect target for anti-CD3 as well. But we started doing that in mouse models. We did in the 2 mouse models of AD, one that's called 3XTG and the other one APP-PS1. For 3XTG, we treated these mice for 6 months or 4 to 6 months, every 3 times a week. And for APP-PS1 for 2 weeks, 3 times a week. And we performed some cognition tests like Y maze or Morris-Water maze. And we found first that nasal anti-CD3, even these AD animals, it induced microglia homeostatic phenotype and reduced MGMD phenotype. So, which is consistent with what we've been seeing in MS as well. And these animals did better in the short and long-term memory measured by Y maze and Morris-Water maze. And what I think is really interesting is that for the first time, we showed that T cells are actually in contact or in close contact or interacting with microglia. This is a CD4 T cells in the anti-CD3 treated group. This is likely another T cell, probably CD8. We're investigating how this is happening, how the cells are getting to the brain, what are the kinetics for the cells to get there. But you can clearly see that, how our hypothesis that these T cells are migrating to the brain and interacting directly with microglia. It's probably what's causing this change in the modulation. So there's several things we're doing now to understand this interaction. And so, as I mentioned, a phase 2A study would start with 8 patients, and the outcomes are going to be measured: the safety, microglial PET imaging, serum biomarkers, and immunological studies. So, are planning to start pretty soon. We also have some data on ALS. Again, we treated these mice in SOD mutant mice for 3 times a week for a couple of months. I'm not going to go too much in details because of time, but again, we see the same pattern of microglia activation. I mean, anti-CD3 reduce this activation of microglia and improves the loss of body mass, and also improves rot-to-rot performance. It's a motor skill, and most importantly, increased survival. So we are now also investigating further this anti-CD3 as we speak in SOD ALS, and trying to understand better, more in details what's happening. And there's also this beautiful paper that we are ready to submit. That was led by Saif Easy. We're investigating nasal anti-CD3 in traumatic brain injury. So it's a beautiful paper. Hopefully it's going to come out soon, in which we found that nasal treatment actually improved memory, improved motor functions, and several other parameters that were tested in this model. And even the size of the lesion, as you can see here, was reduced by anti-CD3, so which is really striking, as you can see here. So we are investigating how this is working, but again, it has to do with the microglia. There is less activated microglia, less infiltrating monocytes. And again, this is just to show that in several time points where we see it's a reduction in inflammatory microglia signature, most likely having this homeostatic microglia instead of this MGMD or DAM microglia that occurs when there is this TBI. And here we're seeing that, again, the anti-CD3 is working by IL-10, as Lior showed in his paper in 2016 for EAE. And it seems that IL-10 is also driving this effect of microglia. So with that, I'd like to conclude saying that nasal anti-CD3 induced Tregs and dampens activated microglia and other inflammatory process. In nasal anti-CD3 is safe and applicable to multiple neurodegenerative disease in which microglia activation plays a role. So I don't have a acknowledgement slide here because there's so many people involved in this over 30 years of study. But, Howard first is the person who we need to thank for all these amazing, tests and ideas that he had. So, and I thank you once again for the invitation.
Rafa, thank you very much. Beautiful talk. And you've been involved in all the things we're doing, all the basic immunology, all the work you've done, all the years on oral tolerance. What do you think about the dosage, and how do we find the right dosage?
Well, I think we had already a good idea by looking at immunologic effects in healthy individuals. It's tough to, for mouse, we did it several times, and the best dose is usually 1 to 10 microgram when you give nasally. And for humans, it seems to be 50 microgram. So I think the dosing is pretty much what, it's not going to be too much difference from if you give 50 or 100. But it's indeed a tough question to define. And I think the studies now are going to show us if you're right about this dose or if you need to increase it or not. So, but I think that's the way. We already have a clue that it's 50 micrograms.
I have another question to ask you. It's interesting when you give the nasal anti-CD3 and you induce these regulatory cells, the anti-CD3 is not antigen specific, but it is inducing cells that go to the brain or go to other organs. Where do you think the specificity comes, or are the cells going everywhere?
I think it's actually going everywhere. And once it gets to the point where it's inflamed, because when there is an inflamed tissue, there is signaling attracting these cells. So the cells are not going to be induced based on a specific antigen, as you said. But if you have a brain inflammation, so they're going to be prone to go there because there are chemokines being secreted. And once they get there, so then they, what I think in a lay way to say, they know what to do. And they interact with these other cells that are inflamed and producing these cytokines like IL-10, and that's how I believe they regulate and they promote. I mean, the mechanism of action is.
Now, in the original paper by Lior, which Francisco participated in, we saw changes both in astrocytes and microglia by the nasal anti-CD3. We don't know. Do you think it's affecting both cells at once, or maybe affecting one cell that then communicates with the other? Because Fran shows that they can communicate.
I think that's pretty much it. Based on what Fran and Oleg showed, it's such a complex environment. So I believe that anti-CD3 is producing IL-10, and IL-10 is able to modulate both cells. But then once it starts modulating microglia or astrocytes, one is interacting with the other as well, and then it comes the whole thing. So which comes first, I don't know, but I think the key point is IL-10.
All right, thank you very much, Rafa. Nice presentation, and I know you're working very hard now on you just got a grant to do the nasal anti-CD3 and ALS, and you showed the nice data on Alzheimer's. Thank you very much.
Thank you.
Okay, I'm going to continue with the seminar, and I will finish up and talk about our work on Protollin. So let me show my slides. Can everybody see it?
Howard is not showing yet.
All right, Fran is helping. Yep.
Yeah, we got it.
Okay, so thank you very much. Thanks to Fran, thanks to Oleg, thanks to Rafa. I'm going to finish by talking about another way we're working on innate immunity. And this is by the induction of phagocytic monocytes by a proteasome-based adjuvant for the treatment of Alzheimer's disease. I'll show you a lot of basic work and then tell you a little bit about our human trials. So Oleg spoke about Alzheimer's disease, and there's different aspects of the innate immune system that can affect Alzheimer's disease. Oleg talked about the different microglia. We also want to modulate the innate immune system. And one way is by giving a monoclonal antibody that affects microglial function. And this is something that Rafa just spoke about. And I'm going to talk about Protollin to affect peripheral innate immunity that then could affect monocytes that go to the brain. Now, how did this all start? A number of years ago, there was a big trial where AD patients were immunized with A beta, because it was shown that if you immunize animals with A beta, you get anti-A beta antibodies, and it clears disease. And so they treated Alzheimer's patients. But unfortunately, 6% of the AD patients that were immunized with A beta in an adjuvant developed a meningoencephalitis that looked a little bit like EAE. And that led us to ask a question, well, if these people got something that looked like EAE, what if we immunized APP mice? Are they more susceptible to EAE? And could you induce an EAE by giving beta amyloid with pertussis? So just as a background work that Alon Monsonego did in the lab, we found that there was increased T-cell reactivity to amyloid protein in older humans, and that's probably the reason you got this encephalitis. Now, the experiment didn't work. When we immunized APP animals with MOG-CFA, the disease they got was similar. And if you immunized with A beta and CFA, there was no disease. However, interestingly, when we looked at these Alzheimer's animals that were given EAE, the animals with EAE had clearance of the A beta. So giving an A beta animal EAE cleared the A beta. Now you can't treat, well, first of all, the question was, how does this work? And one question was, is it antibody dependent? Because antibodies were shown to be very important to clear A beta. And although we couldn't find A beta antibodies, we did an experiment where we induced EAE in B-cell deficient APP mice. So they couldn't make antibody. And as you can see, whether you immunized, when you immunized with MOG and gave EAE in the regular mice or with the B-cell deficient mice, you cleared the A beta. So this was not an antibody-mediated event. So then the question is, can one induce A beta clearance without inducing EAE? Can't treat Alzheimer's patients by giving them MS. Interesting, there's a suggestion that people with MS may get less Alzheimer's. So after a lot of work, we found a adjuvant called Protollin, which is a proteasome-based adjuvant, and used it to clear amyloid deposition in a mouse model of amyloid. Now, this is the adjuvant. It's a mixture of LPS from Shigella and Neisseria outer membrane protein, and then you get Protollin. And this Protollin is a TLR2 and a TLR4 agonist, so it stimulates these innate cells. And Danny Frankel, who was in the lab, did an experiment where he gave nasal Protollin with glutaminyl acetate, which was used for the treatment of MS, and it worked. But then he did another experiment where he just gave the adjuvant alone. And this adjuvant worked very well to prevent amyloid deposition. And here's the experiment. Here's an animal where he gave 5-month-old mice. He was treated weekly for 8 months with the Protollin. And you can see in the animals that were treated, there was a marked reduction in the A beta. And even if you take older mice, 24-month-old mice, and treat for 6 weeks, you also saw a reduction. Other work that he did is he gave the Protollin intranasally for 6 weeks, and then was able to show phagocytosis of A beta by microglia in vitro, and this is using this N9 line. And here he put microglial cells plated on the brain section, gave the Protollin, and you could see that you phagocytized the A beta. We continued to work on this, and this is work that Oleg Butovsky did, where he created CX3-CR1 GFAP mice so that we could see the effect of Protollin on peripheral monocytes. So he created these mice, and then we treated them with nasal Protollin, given 3 times a week or given longer, this is group 1 and group 2, to see whether it affected A beta and whether monocytes came into the brain. And indeed, you could see the recruitment of monocytes into the brain. You can see here in the treated animals, the monocytes here are green. And here you see it with the proteasome, more monocytes. You can see more monocytes with the protollin and a decrease of A beta in these animals. Now, when we did this, we looked at activation of different cells in the mice given protollin. And since this is a TLR2 and TLR4 agonist, it could affect scavenger receptors. And here you could see that there was activation of SRA positive cells, which are scavenger receptor cells, in mice given a nasal protollin. Dan Frankel and us then collaborated with Joel Khoury, and he found that SCARA-A deficiency impaired the clearance of soluble A beta by mononuclear phagocytes and accelerates Alzheimer's-like disease progression. So this identifies mononuclear phagocytes in the periphery as being important to go to the brain and clear A beta, and is the theory for giving people protollin to activate monocytes in the periphery. When Danny Frankel went back to Israel, he did some studies in a cerebrovascular amyloid model. And one of the other aspects of amyloid type disease is CAA. And in these particular experiments, he found that nasal protollin reduced cerebrovascular amyloid deposition. And you can see it here. There's a decreased number of the vessels that have the amyloid. And this is important because in people treated with aducanumab and some of the other monoclonal antibodies against amyloid, you get ARIA, or deposition and inflammation associated with the vessels. And this is important because the protollin doesn't do that. So the working hypothesis then is that nasal protollin induces the activation, recruitment of peripheral monocytes to the brains of mice where they clear A beta. So we've continued now to study this, and I'll show you, and then I'll tell you about some of our human studies. And this is work done by Yoda Polikepti in the laboratory. And she did some work using the APP-PS1 mouse model that develops amyloid deposition and cognitive impairment at 6 months. And the first experiment that she did was to give nasal protollin and see how it affects peripheral LY6 monocytes. And here you can see she's treating it every other day for about 14 days, and then looked at the cervical lymph nodes and the spleen and the bone marrow. And as you can see, nasal protollin increases the frequency of LY6 monocytes in the spleen and bone marrow of these mice. Here you can see cervical lymph nodes was not significant, but in the spleen and the bone marrow, you had a marked increase of LY6 monocytes. And then we did RNA-seq to look at the transcriptional changes in cervical lymph node LY6 high monocytes. And you can see there were a number of genes that were upregulated by nasal protollin. And over here you can see C1qa, SPO1, AXL, all upregulated in these monocytes, showing the activation of these cells. It also induces transcriptional changes in the splenic monocytes. So that was the cervical lymph node monocytes. And here you have the changes in the splenic monocytes. And it also, interestingly, induced transcriptional changes in bone marrow LY6 high monocytes. So this suggests that when you give nasal protollin, you're not only expecting cervical nodes and splenic nodes, but you're actually beginning to affect bone marrow monocytes, whether that's, we have no evidence that the protollin gets into the bloodstream, although we're checking that, but it may be migration of these cells into the bone marrow. And then if you stimulate bone marrow monocytes against A beta 1 to 42 in vitro, it actually increases the ability of these cells to phagocytose A beta. And you can see that treatment with Protollin increased the ability of these bone marrow monocytes to phagocytose A beta. Then looked at the recruitment of these cells into the brains. This is very important because if you want to treat Alzheimer's patients, you want to recruit peripheral monocytes into the brain. And in these experiments, Yoda treated animals over 14 days and then looked at CD4 high expressing cells in brain cell suppression. And you can see an increased number of monocytes. You see it over here, an increased number of monocytes in the brains of these animals that were treated with Protollin. And here you see the significance. Here there's also a little bit with the dendritic cells, not with CD3 cells. So it's an innate stimulus. Now, a very interesting experiment is that Yoda then looked at what happens to microglia from APP PS1 mice treated with Protollin. And although Protollin affects peripheral monocytes, it can affect microglial cells. It affects innate cells in both places. And many of the important genes, many of them MGND genes, were downregulated, ApoE, CLEG7, AXL, etc. And so that this shows that you can affect microglial cells by monocytes that go into the brain. And it does induce these transcriptional changes, and this is just a summary of all the different changes that one has. Oleg just presented this, and these genes include the MGND type genes. We don't know exactly which genes are more important, but the message is that the nasal protollin affects monocytes that go into the brain, and those monocytes in turn can affect microglial cells. Now, Yoda is looking at the possibility that some of the protollin might get into the brain and directly stimulate the microglia. But in Danny Frankel's experiments, he never found that the protollin got into the brain, but we have a new assay to check that. So what about the effect of nasal protollin on total brain cells? This is an interesting question. So in this experiment, Yoda gave nasal protollin again for 14 days. And we did single cell RNA-seq on the brain cortex and hippocampus. Now, we think that the cortex is more important because it is where most of the plaques in the disease is. So here's the single cell RNA-seq, and you can see all the different clusters. And these are the clusters in the animals that were treated with PBS. And these are the clusters of the animals that were treated with Protollin, and this is in the cortex. And what's interesting is that there's two clusters that change in the animals that were treated with the Protollin, and that's cluster 1. And here is cluster number 9. So we then used various gene expression to identify which cells are microglial cells and which cells are astrocytes. And we used for the microglial cluster, we used CXCR1, CSFR1, and these genes to identify microglial clusters. We then also identified astrocyte clusters, including CGA1, GRIA1, et cetera. So that's how we identified the astrocyte clusters. And then we also looked at gene expression in monocytes versus myeloid suppressor cells so we could see where the nasal protollin was having an effect. And returning now to the major figure and looking at number 9 and number 1, which changed. And in cluster 1, these are the genes that we found. This identifies the genes that show that these are microglia or astrocytes. And in cluster 1, we identified genes that are increased in the microglial cluster. So that's going to be very interesting to understand which microglia change. Now, then this is another way to look at this single cell data. And this is percentage of cells in the individual groups. So this is the cortex, the control animals. And this is the Protollin-treated animals. And you can see that in Protollin-treated animals here in blue, cluster 1, you have a marked increase in cluster 1. You also have an increase in choroid epithelial cells, somewhat of a decrease in the MGND microglia. These are the major changes. Now, in the hippocampus, where there weren't as strong effects, you found that the choroid plexus epithelial cells were changed. But this is the very interesting data to understand what's going on in cluster 1 and in these choroid plexus epithelial cells. Now, if this is true, can monocytes that are treated adoptively, can you adoptively transfer them and affect the behavior of APPPS1 mice? So that would mean that the monocytes are doing something, and it isn't just the Protollin that's going there. So what Yoda did is she took animals and treated them with nasal Protollin or PBS, took the monocytes and then transferred them into APPPS1 mice and followed them over time. You can see how long of an experiment this was. You followed the animals for 8 months. First of all, she didn't find that the adoptive transfer affected the movement of the APPPS1 mice, but it did improve the behavioral deficits. And here you can see the animals treated with the Protollin, the distance in the novel arm changed, as did compared to the wild type. So it did affect the behavioral properties, and it improved the behavioral deficits. And here you see it again, wild type mice and the patients treated with the Protollin improved more towards the normal. So it affected behavior. And it also reduced the level of A beta in the brains of animals. And this is shown right here with the Protollin-treated animals and the decrease in A beta. Now, the next big question is, can we affect what happens if you give Protollin to human PBMCs? And we did this in a healthy individual. Now we're giving Protollin to Alzheimer's patients now. And it's going to be very interesting to see how that affects the monocytes. But here's an initial experiment of single-cell RNA-seq of human PBMC monocytes from a healthy individual. And here you can see the untreated monocytes in the RNA-seq. And over here you see the Protollin-treated monocytes. And it looks like there's a new cluster that appears, that this is in vitro, that the Protollin-treated monocytes is a new type of cluster that we're inducing when we treat peripheral monocytes with Protollin in vitro. Now, the other thing we're doing now is taking AD monocytes in vitro and treating them with Protollin to see how they change. There is data suggesting that peripheral monocytes from Alzheimer's patients are defective in their ability to phagocytose A beta. So we want to see whether that can change. So this is many years' work. And of course, it ultimately leads to a clinical trial. And we just completed a phase 1 single ascending dose trial in mild AD. and here at the Brigham and Women's Hospital. And what we did is we gave 0.1, 0.5, 1 milligram or 1.5 milligram of Protollin. It was given nasally on day 1 and on day 14. And then we measured safety and immune responses. We would have rather given it weekly or maybe even 3 times a week, but the FDA wouldn't let us. They said, first you have to show safety because it was only given on day 1 and day 14 before in Shigella and influenza vaccination. Actually, we were very lucky that they allowed us to go to AD patients as opposed to healthy controls. So I'm happy to report that the trial is completed. There were no major side effects, even at the higher doses. We saw some effect at the 0.1 and 0.5, although not dramatic, but we're beginning to see effects at the 1 milligram, and we'll know about at the 1.5 milligram dose. After that, we'll do another study in AD patients where we treat, instead of on day 1 and 14, treat on day once a week, as Danny Frankel did, or treat 3 times a week to induce these responses. And then all the data that we have on the peripheral monocytes and on the immune changes, we can measure in these AD patients. And once we get the regimen and dose done, we're ready to do a trial where we will have a placebo-controlled trial of nasal Protollin, where we will measure effective A beta clearance and also effect on cognition. Now, one of the interesting things that everybody knows, the controversy about clearing A beta, and does that work in Alzheimer's? The aducanumab trial was partially successful but not approved. At the CTAD meeting, which is coming up in San Francisco, we look forward to results of new trials with anti-A beta antibody. We know that A beta triggers tau, and tau is what causes cognition deficits. And if the nasal Protollin works and clears A beta, it can be given early. It could even be, and is safe, it could be given to prophylactically to people in their 50s or 60s who are beginning to have evidence of some A beta accumulation, even though they don't have any cognitive function problems. So this returns to the initial slide, innate immunity in Alzheimer's disease. If we talk about Alzheimer's being A beta and then tau and then microglial activation, the monoclonal antibody that Rafa talked about could be given later in Alzheimer's in terms of clearing, in terms of restoring microglia. And the Protollin could be given earlier to induce recruitment of LY60 high CCR2 monocytes to A beta plaques, which could then clear the plaques. Obviously, these treatments could be given together, and we look forward to moving this forward for the treatment of Alzheimer's disease. A lot of people worked on this. The initial work was done by Danny Frankel. Oleg did a lot of work on this. We had help from Dennis Selko and his group. But the main person whose data I showed more recently, who's really carrying the project, is Yoda, who's doing an outstanding job in all of the immunology and defining this whole area. So I want to thank you very much, and it's been my pleasure to present the this seminar of innate immunity in neurologic diseases from our center for the Prada Symposium. We think the innate immune system is very important. And we have a wonderful team of people working on it. Oleg, Fran, Rafa, myself, Yoda. Another area that I didn't speak about relating to innate immunity is the microbiome, and Lori Cox is working on that. So I want to thank the Prada Foundation. We have 2 or 3 minutes left, so I'll turn Rafa or Oleg, do you want to ask me any questions or any closing comments?
So, Albert, I know you do a lot of therapeutic translation, which is what we need to do.
There's Fran too. Hi, Fran.
Right. When do you think, what would be the first treatment on your side and when it's going to happen? It's a tough question, I know, but...
Or what, for the Protollin or for the anti-CD3?
Either one of them, yeah.
I think the anti-CD3, we're doing it now in MS and we show it. And I think in next year we'll do a placebo trial. And hopefully next year we'll do an initial trial of the anti-CD3 and Alzheimer's as well. The Protollin, if once we get the dose next year, we should do it.
So Howard, in terms of biomarkers for to quantify the outcome, right, of treatment with anti-CD3, which one do you think is the most exciting? Because that's obviously it's a challenge when we think about progressive MS, for example.
Yeah, I think the most exciting biomarker is PET imaging because it shows the activation in the brain. And I think that's going to be very good for your studies, Fran, where you target astrocytes, or Oleg, where he targets microglia. And I think we're very lucky to have PET imaging. And I would encourage all the people who are watching that if you want to study innate immunity in the brain in vivo in people, microglial PET imaging is very important. All right, we're at the end of our time. Again, thanks to Fran, thanks to Oleg, thanks to Rafa, thanks to all our team, thanks to the Prada Foundation, and we look forward to more exciting science. Bye.
Bye-bye.